Yes, good morning. Good morning, students. Our next topic, double fertilization in angiosperms. If you recall, in a last lecture, we talked about artificial hybridization. We move to the next topic, double fertilization. At the end of the pollen-pistil interaction, we studied that the pollen tube enters the ovule through micropyle and further enters inside the embryo sac, guided by the filiform apparatus, and finally entering in any one of the synergids out of the two, any one synergid in which the pollen tube shall enter. After entering in one of the synergids, the tip of the pollen tube is ruptured and it releases two male gametes. It releases two male gametes. Obviously haploid male gametes. Inside the cytoplasm of synergids, as you can see, this degenerated synergid in its cytoplasm, two male gametes are discharged. One of the male gamete that is moving towards the egg cell, this one. One of the male gamete moves towards the egg cell. Here it's gonna fuse with the egg cell, the nucleus, and will complete the syngamy. It will complete the syngamy. Syngamy is the fertilization. Syngamy is the process called fertilization. So syngamy will result in the formation of zygote, a diploid cell formed through the fusion of one male gamete, the haploid male gamete, and another cell, the haploid egg cell. That's the female gamete. Their fertilization is called as syngamy. Actually, it's the synonym of fertilization. And the product formed, that is termed as zygote. This is the diploid cell. This is the diploid cell. On the other hand, another male gamete that was discharged in the synergid, it will be moving towards the central region of the embryo sac, where the two polar nuclei are present within the central cell. Inside the large central cell, centrally placed two polar nuclei, each one haploid. One of the male gamete reaches to that and carries out fusion with that. The process is identified as triple fusion. It is known as triple fusion. Product is called as primary endosperm nucleus. The ploidy of the same, the ploidy of this structure, primary endosperm nucleus, its ploidy is 3N. Reason: one haploid male gamete and two haploid polar nuclei. Collectively, it will make 2N plus N. That's gonna be the 3N. As three nuclei are involved in this fusion, the process is identified as triple fusion. Process is identified as triple fusion. You could see that inside the angiosperms at the time of fertilization, two different types of fusions occurred. One was syngamy, which led to the formation of zygote, one male gamete and one egg cell. And another one that is triple fusion formed by the or carried out by the one haploid male gamete and two polar nuclei, leading to the formation of primary endosperm nucleus, PEC, triploid structure. As within the embryo sac, two types of fusions are occurring, syngamy as well as triple fusion. This is the reason why the phenomenon of fertilization is called as double fertilization in case of angiosperms. In case of angiosperms. Now, if you compare it with the gymnosperms, it's gonna be the single fertilization. They do not have a endosperm formation after the fertilization. Their endosperm is pre-fertilized endosperm. So they have only fertilization leading to zygote formation. So that's a single fertilization in gymnosperm. Double fertilization, it's a characteristic feature of only angiospermic group. Remember this. Unique to the flowering plants, this event. Acha, the primary endosperm cell, the nucleus is primary endosperm nucleus, and the whole cell otherwise is a primary endosperm cell. This is supposed to develop endosperm tissue, whereas the other cell that is towards the micropyle, that is termed as zygote, it is supposed to develop an embryo of the seed. Remember, the diploid zygote develops into the diploid embryo. The triploid primary endosperm cell develops into endosperm tissue. This is the process of double fertilization. I rapidly revise and then I'll ask questions. You know that at the end of pollen pistil interaction, the pollen tube extended towards the ovule enters inside the ovule via micropyle, and within an ovule, it will enter inside through the synergid cells of the embryo sac. This is being guided by the filiform apparatus. Filiform apparatus guides the pollen tube to enter in one of the synergid. Obviously, at that stage, synergids degenerate. One more fact, that even antipodals degenerate. Literally, two cells are left inside the embryo sac. One is the haploid egg cell. Another one is the diploid central cell with its two polar nuclei. Within this, the two male gametes are being discharged by the pollen tube. Pollen tube tip ruptures and releases in the two haploid male gametes within the embryo sac. One of the male gametes stays near the micropylar end, fuses with the egg cell, the diploid cell. The process is being identified as syngamy. In a way, fertilization. And the product so formed, that is termed as zygote. This is that zygote, a diploid cell. On the other hand, another male gamete, haploid male gamete, moves towards the central region of embryo sac, where the two polar nuclei are lying within the central cell. They also fertilize it. they also get fertilized by it, the male gamete. Because three nuclei are now undergoing fertilization, it is known as triple fusion. The product formed, that is termed as primary endosperm nucleus, a triploid structure. Obviously, the central cell is now converted into primary endosperm cell. Since within the embryo sac, two types of fusions occurred. Number one, syngamy. Number two, triple fusion. This is how the angiospermic fertilization process is identified as double fertilization. This is known as double fertilization. It's a characteristic for these flowering plants, unique for these flowering plants. Nowhere in the plant world you can see this. It's only in angiosperms. The two products of this, one of them zygote and another one primary endosperm cell, respectively give rise to embryo and the endosperm tissue. The zygote gives rise to embryo in future. That's going to be the diploid structure. Primary endosperm cell altogether gives rise to endosperm tissue in future. This is the triploid one. If you compare this process with the gymnosperms, you realize that in gymnosperms, it's only single fertilization within the embryo sac. Only one, that is the egg cell, needs to undergo fertilization by the male gamete to form the zygote. No other structure that is going to undergo the fertilization within the embryo sac. So that's the first thing. Second thing, the kind of endosperm there and in this angiosperms. Gymnospermic endosperm is pre-fertilized, where, at the time when embryo sac was developing, same time the endosperm was formed over there. And it's the haploid one. Whereas in the case of angiosperms, the endosperm tissue that is developed is, first of all, post-fertilized one. It will be formed after the fertilization, and it's gonna be the triploid one in nature. That's a major difference between the two, gymno and angio. Single fertilization in gymno, double fertilization in angio. Pre-fertilized haploid endosperm in gymno, post-fertilized diploid or a triploid endosperm in the angiosperms. That's all about it. Fine. So let's talk about the double fertilization. The pollen tube enters inside the ovule through micropyle. Correct. Kashish and others. Micropyle is the structure through which pollen tube enters the ovule. Which structure shall guide the pollen tube entry into the embryo sac? Shruti, correct. Kea, correct. Filiform apparatus. It's the filiform apparatus that will guide the pollen tube entry into the embryo sac. Inside the embryo sac, in which structure the pollen tube will open? Devanshi and Kashish. Correct. One of the synergid cells, cytoplasm. Simultaneously, the synergid cell will degenerate, and within its cytoplasm, the pollen tube shall open up its tip and discharge the gametes. How many male gametes are discharged inside? Yes, Duti and others correct. Heli and others correct. Two male gametes are discharged inside the synergid cytoplasm. Meanwhile, what happens to other synergid as well as the antipodal cells? Yes, correct, Nidhi and Priya. Degeneration of these cells, they get degenerated. So literally in an embryo sac, at the time of entry of male gametes, how many cells are left? Tanvi, correct. Two cells. Can I have the names of those two cells? Please provide the names. Astha, correct. Egg cell and central cell. One of the male gamete, haploid male gamete, towards the micropylar end, it will fuse with the nucleus of which cell? Yes, Pari, I mean correct. Egg cell. It's gonna fertilize the egg cell. This fertilization is also identified as which process? Heli correct, Devanshi correct. It's a syngamy. The process is identified as syngamy. And what about the product of that syngamy? Yes, Shruti and others. Zygote. It's gonna be haploid or triploid or diploid or tetraploid. Diploid, correct. Diploid zygote over there towards the micropylar. How about the other male gamete? It's gonna move towards the center of embryo sac to fertilize what? Yes, Heli and Kushi, correct. The two polar nuclei. Each polar nucleus is what? Haploid, diploid, triploid, tetraploid. Haploid. Muhammad Waris and Priya, correct. So two of them, each one haploid, which is a part of a central cell. This fertilization, fusion of this nuclei, the two polar nuclei and one male gamete, that is termed as what? Correct, Pari and Prachi, it's correct answer. Triple fusion. Both this syngamy and triple fusion occurring within the embryo sac. Collectively, this phenomenon is identified as what? Yes, Karuna, Dhyani. It's termed as double fertilization. Which cell is a product of syngamy? Zygote. Correct, Heli and Kia. What is the future of this zygote? It has to develop into what? Kia. Perfect. Embryo, Muhammad Waris. Correct. Embryo, it is supposed to develop into an embryo. And what is the future of primary endosperm cell? Diksha. Correct. Fabulous. Endosperm tissue. The double fertilization is a characteristic of which group of plants? Unique to which group of plants? Diksha, it is wrong. It's wrong to say gymnosperm. It's for angiosperm, the flowering plants. And what shall be the two words to describe the endosperm in angiosperm? Angiospermic endosperm के लिए दो word अगर describe करने हैं. One is triploid, Astha has rightly said. ये क्या सिंगल फर्टिलाइजेशन लिख रहे हो? मेंटली सिक हो आप लोग. क्या पूछा नहीं वो भी लिखोगे क्या? स्वाति is correct. Post fertilized and deployed in nature. ऐसा मैंने पूछा. And what will be the two lines to describe the gymnospermic endosperm tissue? Yes, Nidhi, correct. Tanvi, more appropriate. Pre-fertilized and haploid, that should be the combination for the features of gymnospermic endosperm tissue. What type of ovules are there in gymnosperm? याद है ना, एनाट्रोपस ओव्यूल हमने लर्न किया था एंजियोस्पर्म का वन ऑफ द प्रोमिनेंट कैटेगरी. तो अगर ओव्यूल की पोजीशनिंग के कॉन्टेक्स्ट में अगर टाइप, now what nonsense answer you guys are giving? No, it's wrong answer. Understand the question. I am not saying the covered or uncovered ovule aspect. I am saying if anatropous type of ovule is one of the pattern of ovules seen in angiosperm, which type of ovules are seen in gymnosperm with respect to those patterns? Yes, what is the answer? Against anatropous pattern of angiospermic ovule, dash is the pattern of ovule in case of gymnosperms. Well, this no reply from your side is a clear-cut indicator that how insincere you all are. You have been given one topic for self-study, that was the type of ovules. For NEET exam preparation, I asked you to go through that. But not a single student deserves to be called as a sincere. All are hopeless with respect to the NEET exam preparation. All are insincere, not interested in studies. So this was about fertilization. Naturally, fertilization is to be followed by post-fertilization events, which involves the primary endosperm cell to be developed into endosperm tissue. That zygote needs to be developed into an embryo. Your ovule after the fertilization will turn into what is called as seed. The unfertilized ovule now gets fertilized. It is called as seed. Your ovary after the fertilization is transforming itself into fruit. It ripens up gradually and it turns to enlarged structure and it is called as fruit. And the wall of ovary shall be now called as fruit wall, for which another term is given, that is called as pericarp. So endosperm formation, embryo formation, maturity of ovule into seed, maturity of ovary wall into fruit wall or a pericarp, and maturity of an ovary into the fruit. These are the post fertilization events. There are 5 major events that take place immediately or gradually after the fertilization process. Which are the first ones? First, what shall happen? Yes, endosperm formation is the first such event.
मम्मी
Which
वहाँ
is the
तब से
second
चार्जिंग नहीं था.
post-fertilization event? Embryo development. Third, ovule into seed. Maturity of ovule into seed. Fourth, correct, ovary into fruit. And last, which is the part of the fourth event only, but just for your understanding, ovary wall into fruit wall. What's another name for ovary wall? फ्रूट वॉल के लिए दूसरा नेम क्या है? करेक्ट, पेरिकार्प. Now, first we will talk about endosperm formation and then we will talk about embryo development. Let's talk about endosperm formation. See, the endosperm formation will precede embryo development. Inside the seed, before the zygote develops into an embryo, endosperm tissue has to be ready. It has to be prepared. See, logically, it is appropriate that before the, this, before the embryo is formed, if endosperm is developed, it will be an assurance that nutrition is available for the developing embryo. Developing embryo shall require nutrition. Formation of endosperm before that process of embryo development starts is a guarantee. It's an adaptation to ensure that before the embryo is formed, its nutrition material, that is endosperm, is ready. So this is the reason why endosperm development should precede. It should start first and later on goes on this embryo formation. I hope you have understood. It's an adaptation on the part of angiosperms. They ensure that first they complete the endosperm formation. Then they go for embryo development, because developing embryo will require nourishment, and that can be offered by already prepared endosperm tissue. Coming to the endosperm formation. From which cell the endosperm formation should start? If I ask you, which is the cell that is supposed to give rise to endosperm formation? Primary endosperm cell. The cell I am asking, it should be primary endosperm cell. It is the one involved in the endosperm formation. This is that primary endosperm cell. After the degeneration of other cells, here you have a zygote, and upper one is the primary endosperm cell, and then its nucleus, primary endosperm nucleus. Now the process goes on in this manner. Now, before I go in detail, let me tell you that when a endosperm tissue will be formed, because this cell was triploid, our each cell in the endosperm tissue is also triploid. So it's a triploid endosperm. And it is filled with, the cells of this endosperm are filled with reserve food material that will be used for the nutrition of developing embryo. जब इसमें से embryo develop होगा, तो उसके लिए nourishment provide किया जाएगा. Nourishment will be provided by the cells of the endosperm because they have lots of reserve food material in them. There are different types of endosperm formation: nuclear endosperm, cellular endosperm, halobial endosperm. Again, one more topic for the self-study for NEET exam preparation. Source could be the reference material, any reference material, or a internet. By the way, we have ordered for you the books for a 12th standard reference. Soon they will be provided to you. They are good for state board as well as NEET exam preparation. So now, whenever you will go through those notes or those books, you will get such information. The topic is types of endosperm formation. The type we are going to talk about over here is a nuclear endosperm formation. There are other types such as cellular endosperm formation and halobial endosperm formation. Self-study topic. Coming back to that, the most common type of endosperm formation in angiosperm, that is called nuclear endosperm formation, is the one in which the primary endosperm nucleus, this triploid nucleus, it undergoes repetitive nuclear divisions. No cytokinesis, only continued karyokinesis. This leads to formation of mass of triploid nuclei. This mass of triploid nuclei is called as free nuclear endosperm. For this, the term employed is free nuclear endosperm. Gradually, this nuclei move towards the periphery of embryo sac, and later on the cytoplasmic division occurs. Each one is surrounded by some amount of cytoplasm, and around each one, the cell wall formation takes place like this. And finally, the cellular endosperm also comes into existence. Finally, if you see the coconut, tender coconut, green coconut, the liquid that you see in that, the coconut water, that coconut water is nothing but free nuclear endosperm. And the kernel that you see at the periphery, the white kernel, the malai that you call, surrounding this water, the white color kernel, that is the cellular endosperm. जो लिक्विड होता है वो न्यूक्लियर एंडोस्पर्म और उसका कैर्नल मलाई जो होता है वो सेल्युलर एंडोस्पर्म को रिप्रेजेंट करता है. So that is how the endosperm tissue that has been developed within the seed. बोलिए, कोई डाउट है इसमें? You have any doubts in the same? No? Then let's start straight away the question answer part. Sneha, you are raising the hand. That means you have any question? You have any question? Okay, by mistake. Fine, we can move to the next. Before we go to the next, let's have a revision of the same. See, which activity shall take place first? Embryo formation or endosperm formation? Yes, it's the endosperm formation. Mrunali, correct. Can you justify why should endosperm formation precede embryo formation? Yes, because it is meant to provide nourishment or nutrition to the developing embryo. So if you carry on with the embryo development before even endosperm is formed, embryo will be deprived of nutrition. So system ensures that first the nutritious tissue endosperm is formed. Once it is formed, then only the embryo development starts. This is to ensure the nutrition supply to the developing embryo. This is an adaptation on the part of angiosperms. Which cell forms endosperm tissue? Yes, Diksha and others correct. Some are wrong also. When I ask about cell, you have to say primary endosperm cell. There are two things, a triploid nucleus in the center, and then the surrounding cytoplasm and cell wall collectively makes it primary endosperm cell. I said primary endosperm cell is the one which will develop into the endosperm tissue. Further, next, what is the importance of endosperm? What is the importance of endosperm? Be specific. It has a reserve food material and it will provide nourishment to the developing embryo. That should be the reply. Which are the different patterns of endosperm formation? The different patterns of endosperm formation. Cellular is the one, correct, Devanshi. Nuclear is another one. And yes, Shruti, Swati, etc. Halobial is another one. Nuclear endosperm formation, cellular endosperm formation, halobial endosperm formation patterns are there. One of the most common one, that is the free nuclear endosperm formation. Which nucleus undergoes repetitive divisions? Pen, isn't it? Nidhi and others, Jal and others. Primary endosperm nucleus is the one which will undergo repetitive divisions to form a mass of triploid nuclei without cell wall and cytoplasm around them. This nuclear mass is called as what? Free nuclear endosperm, isn't it, Devanshi? Correct reply. It's called free nuclear endosperm. Large number of nuclei over here. Gradually now this nuclei start developing cell wall around them, gradually transforming the nuclear endosperm into which one? Yes, cellular endosperm. In a green tender coconut, the coconut water that we drink, that represents what? That represents what? Free nuclear endosperm. And surrounding this water, the white kernel that is available, it represents what? Yes, one with another's. Correct, Sneha. It's a cellular endosperm. See, based on the endosperm utility, there are two types of seeds. For example, on a one hand, you have a non-endospermic seeds. The one in which endosperm may be completely consumed by the developing embryo. Endosperm will be completely get consumed by the developing embryo even before seed is mature. So matured seed will be without endosperm tissue. We call them as non-endospermic seeds, X-albuminous seeds. Example, amongst the dicots, which is, most of the dicots are non-endospermic, pea, bean, groundnut, etc. will come under this category. Very few, monocot plants, they are having non-endospermic seeds. One of them is orchids. One of them is orchids. On the other hand, you have a endospermic seeds, the one in which endosperm persists even in a mature seed, which means what? The developing embryo must have taken up this certain amount of endosperm, but it is not fully utilized. So even in a mature seed, one can see the presence of endosperm. That's called endospermic seeds. Very few dicots are having endospermic seeds. Example one, exceptional such case, that is castor. Most of the monocot plants are having endospermic seeds like in maize, wheat, rice, jowar, barley, etc. So these are the two types of seeds based on endosperm availability in a mature. These are the mature seeds based on the endosperm availability. These are the two different types over there. Which are the two different types of seeds based on availability of endosperm in the mature seed? Which two? Okay. I ask you, which is an example of a dicot that shows endospermic seed? What's your answer? Diksha. Correct. Castor is the one. In which monocot plants non-endospermic seeds are present? Heli, correct. Orchids. Name few dicots which are having non-endospermic seeds. Heli, correct again. Pea, bean, groundnut, cashew, almond, etc. Name few monocot plants with endospermic seeds. Maize, rice, wheat, etc. Fine. We have a break for 5 minutes, 5 to 6 minutes break. Then we meet again and continue with our next topic, embryo development. Mindya ma'am, hold the recording for a while. So, in a previous topic, we talked about endosperm formation. We move to the next one, embryo development, embryo formation. We know the fact that embryo development will take place from the diploid cell called zygote, formed towards micropyle, and will take place only if endosperm is already formed. Then after only it is gonna be taking place. So important aspect to note here is it's a embryo development from the single cell called zygote, and it's gonna succeed only the endosperm formation. Once it is done, then only it is going to take place. Most of the time, the zygote will divide only once the certain amount of endosperm is formed, as we said that it's an assurance, or it's an adaptation rather, to provide the assured nutrition to the developing embryo. The pattern of development of embryo differs in plants in later stages. In early stages of embryo development, if you see, there is some kind of a similarity. We have a monocot plants, we have a dicot plants also. Look at the dicot seeds and monocot seeds, so much different from each other. But the early stages of development, if you see, before they turn into the mature seeds, at early levels, at early stages, there is a tremendous similarity in them. They are so much similar to each other in the pattern of the embryo formation. Gradually, at the later stages, there are a lot of changes taking place over there, and they become different from each other. Particularly the one that is shown over here indicates the embryo development in the case of dicot plants. Actually, Capsella plant was the one in which such a development was studied. So it's a Capsella type of development of embryo. Capsella was a dicot plant, the first one in which a detailed study of embryo development was done. So the diagram indicates, or it depicts, Capsella type of embryo development. The whole embryonic development is been divided into different stages through which the embryo has to go through. This includes the single cell embryo, that is zygote. Then you have a four-celled embryo, almost T-shaped embryo. This is called as proembryo. Then further it develops into a little larger structure called as the globular stage of an embryo. Subsequent one, heart stage of embryo. Next to that, mature stage of an embryo. These are the different stages of an embryo. Well, one is exam point of view, just diagrams are important. In the diagrams, stages may be asked and you have to identify the stage. Second possibility is to draw the stages of the embryo development. That could be the second possible question related to this topic. As far as details of this process are concerned, how the zygote becomes proembryo and how that transforms into globular stage and then heart-shaped embryo and subsequently the mature embryo with the different parts which are labeled over here. I guess this is out of syllabus and not likely to be asked. So I recommend it will not be asked, so no need to prepare it in detail. Otherwise, you have certain labelings also there. For example, this structure is called as a suspensor. This cell is called as a basal cell. These are the embryonic cells. This was a basal cell. This is an apical cell. Basal cell doesn't divide further. It remains as it is at one stage. And then after the apical cell, it will transform into an embryo over there. So all those things will take place, but you don't need to remember these stuff. I can explain, for example, in globular stage, upper part and lower part. There are two parts formed over there in the clusters of cell. This is called as upper octant, that is towards chelaza, and this is lower octant of cells, embryonic cells, that is towards the uh, micropylar end. This is chelaza octant, this is micropylar octant. This will make certain parts of the embryo. This will make certain parts of the embryo. All that detail is there, but I believe that in board exam, only two possibilities. Number one, the diagrams to be prepared, and number two, the labeling on them, the stages names. That's it. As far as NEET exams concerned, sequence and names, nothing more than that. So that much focus you should pay upon this particular topic. Well, let me ask and revise this. Fine. So let's have this conversation. Which cell will develop into an embryo? Zygote, correct. Is it haploid or a diploid? Diploid, correct. Is it towards center of embryo sac, chelaza end, or micropylar end? Yes, at the micropylar end. Is it before endosperm formation or after endosperm formation? After endosperm formation. What is a term for embryo development? One term that is used to understand the embryo development. एक term है equivalent to the embryo development. Embryogeny or embryogenesis. If you compare the embryogeny of dicots and monocots, at initial level there is a great similarity. At later stages there is a tremendous differences also towards the maturity. Normally, I mean the one which is shown in the diagram, it belongs to which plant's development? This was a typical embryonic development of which plant amongst the dicot, which was the particular case that was studied here? Name of that plant? Yes, Hussain, Wali and Tanvi. Capsella. Capsella type of embryo development is the one that you are looking at. I point out the stage, you have to let me know what is this stage known as. Yes, that's called zygote. Which stage is this next one I am talking about? Yes, that's pro-embryo stage. Which stage is this one you are looking at? Globular stage. Which stage is this one? Heart-shaped stage. How about this one? Last one. Mature embryo stage. What about this structure? Suspensor. How about this structure? Correct, radical. What about this? Plumule. How about this too? Hmm. So that was about the embryo development. Now we shall see the two different types of embryos: dicot mature, mature dicot embryo, and a mature monocot embryo in a grass, particularly monocot embryo. Madam, one or two, Nishil is trying to enter. I'm giving entry. You ask him what is his problem.
Yes,
Nishil,
Nishil Patel.
पूछो.
Nishil, unmute yourself. Nishil Patel.
એને બોલો સ્ક્રીન પર આવે વડીલને.
Nishil, come to the screen. Switch on your video. સર એ લાગતો જ નથી હોય એવું લાગતું નથી ત્યાં.
Nishil, you have to
Patel.
give reply either in a come to the screen or through mic, unmute yourself and speak, or at least in a text form in a chat box you can reply, show your presence. Ma'am, we have to remove him as he is not responding. That means he may be absent physically over there.
Okay, sir.
We got to remove him. Students, in every lecture it happens that one or two students we need to remove for not responding. You guys don't respond by even unmuting or not by texting us. How do we understand that you are physically present over there? You are not switching on your screen, no reply in a text form or in a voice form. So how would we understand that you are present over there? That much cooperation we expect from you. In every lecture it happens with one or two students, and then you are forcing us to remove yourself as if you want us to remove you. It looks like that. Otherwise, how much it takes to write down that, sir, I am present. Tell me, is it difficult to show your presence on the in the meet? Is it really difficult? Please give reply. There are three formats available. You can come to the screen. If it is not working, you can unmute yourself. Otherwise, at least you can say in a chat box that, sir, I am present, some difficulties in my computer. At least that much cooperation we expect from you. You don't follow us in the other aspects. You don't study the things as the way we guide you. You never complete the homework. You never prepare yourself properly. All these things we tolerate. But how can we tolerate that you are not interested in showing your even presence therein? That much cooperation we expect from you. Now, what was the topic? I don't remember now even. Kya tha? Yeah, the stages of embryo development and all that stuff. Well, we move to the next part, that is, just hold on, I will use another diagram. Madam, hold the recording for a while. I need some time. So our next topic is mature seeds of or mature embryos of two different types. One is dicot. So let's talk about mature embryo in dicots. A typical embryo of a dicot consists of an embryonal axis, an embryo axis. See, this is the diagram given in the textbook. But believe me, it is really not clear what it shows. So I have used another diagram to make you understand that what is an embryo axis. Now this is an embryo axis of a dicot embryo. In this embryo axis, along with this, there are two cotyledons. There are two cotyledons. This is the one, this is another one. The two cotyledons enclose the embryo axis. A mature seed is equal to an embryo axis and two cotyledons. When you talk about an embryo axis, it has got following components. For example, the portion of embryo axis just above the level of cotyledon. Now, as you can make out that if this is the embryo axis, the cotyledons are attached to it at a certain position. This one. Now, the region of embryo axis just above the level of cotyledon is this one. It is termed as epicotyle. This is termed as epicotyle. It's going to terminate into the next structure at the tip, and that is called as plumule. This is basically a shoot tip, stem tip. इसको shoot tip या stem tip कहते हैं. अच्छा, the region of embryo axis just below the level of cotyledons. The cylindrical portion of the embryo axis below the level of cotyledon, that one is termed as hypocotyle. That one is termed as hypocotyle. And it's gonna terminate into the last structure that is called as radical. That is named as radical. Achha, this radical will have ultimately, the root tip will have ultimately a covering that is called as root cap. You can see it prominently in this case. So this is about the structure of a typical dicot embryo. An embryo axis with two cotyledons. An embryo axis with two cotyledons. Inside the embryo axis, point of attachment of cotyledon, if you focus where the embryo axis to which the cotyledons are attached. Region just above it, portion of embryo axis just above it, called epicotyle, and it's going to terminate into the stem tip, which is termed as plumule. Then the portion of embryo axis just below the cotyledon attachment to the embryo axis is termed as hypocotyle. It's going to terminate into the part of embryo axis which is called as radical or a root tip. This radical or a root tip tends to remain covered with root cap. Identify. What is this? Mature dicot embryo. Next. In a mature embryo of the dicots, which two structures can be seen mainly? First is the cotyledons, two cotyledons, and second thing is an embryo axis. A mature dicot embryo carries two things, an embryo axis and the two cotyledons. The two cotyledons are attached to the embryo axis. In this, what is an epicotyle? What is an epicotyle? The part of axis above cotyledons, above the levels of cotyledons. Levels of cotyledons means point where cotyledons are attached. And epicotyle shall terminate into what? Plumule. Correct. Another name of plumule? Stem tip. What is hypocotyle? The part of the embryo axis below the cotyledon levels is called hypocotyle. Epi means above, so hypo means below. It's gonna terminate into what along with its another name? Yes, radical or root tip. Which is gonna be covered with what? Yes, exactly. It's gonna be covered with root cap. Last one. Monocotyledonous embryo. Example, embryo of grass. Here is the longitudinal section of the embryo of grass, a typical monocot. They have a single cotyledon, first of all. In monocots, they have a single cotyledon. This single cotyledon is known as scutellum. सुना होगा आपने scutellum term 11th में अगर याद हो तो. So in a grass family, the cotyledon is called scutellum, situated towards one side, the lateral side of this embryonal axis. This much is the embryonal axis, which is the main part of the embryo. As usual, it has got the shoot tip, that is a plumule, and root tip, that is the radical, that is a typical feature. Additional feature over here that you can see that at a lower end, the radical and root cap are covered with the protective sheath. That protective sheath is termed as coleorhiza. and plumule, the shoot apex, remains protected in protective sheath called as coleoptile. Now, this is something which was not seen in the embryo of dicot. So it's a characteristic of monocot embryo that shoot apex will be covered with coleoptile, root tip will be covered with coleorhiza. These are basically undifferentiated protective sheaths. Undifferentiated protective sheath which encloses the root tip and radicle called coleorhiza, and the one that's going to protect the epicotyle that is called as coleoptile. The epicotyle having a shoot apex and a leaf primordia. लीफ प्राइमोडिया क्या होता है कि जिस स्ट्रक्चर में से लीफ डेवलप होने वाला है वो इनिशियल सेल मास को लीफ प्राइमोडिया कहते हैं. So that cell mass is enclosed in a hollow foliar structure known as coleoptile. That is about the monocot embryo. Actually, while making this diagram, a mistake was committed here. So that's why this has been cancelled. You, whenever you prepare, you can write down coleorhiza over here. So that is the one. With this, I conclude it. Which are the additional features in a mature monocot embryo, like in case of grass? The features that are not to be seen in a dicot. Which structures are there? Yes, coleoptile and coleorhiza are the ones. Coleoptile is a hollow foliar structure which encloses which two structures? Shoot apex and leaf primordia together making epicotyle, and together on the upper part of epicotyle rather. So epicotyle has a plumule. This plumule is formed by shoot apex and leaf primordia, and it remains enclosed in a hollow foliar structure. Foliar means leaf-like structure, that is termed as coleoptile. What about the radicle and root cap? Their covering is called what, and what type of structure is that? Yes, that's called coleorhiza. An undifferentiated sheath called coleorhiza covering up this radicle and root cap. Well, last few topics left. Ma'am, you can wind up with the recording. With this, the topic is over. Stop recording.